Dear students, welcome to EPG Pathshala. I am Dr. Rajkishore Sharma, an Associate Professor from Chemistry Department, Delhi University. Today, we are going to discuss about the module Analysis of Energy Dispersive X-ray Spectroscopy under the paper of Surface Analytical Techniques 2. As the name suggests, analysis means detailed examination of elements or structures of a material. Similarly, now in this module, we will study about how to interpret the data obtained from an EDX spectrometer. In this module, we are going through the following topics. Number 1, the analysis, then sample preparation, data interpretation, qualitative analysis, quantitative analysis, elemental mapping, line profile analysis, window type, and lastly, the graph interpretation. So, let's start with analysis part. EDX is a qualitative and quantitative X-ray analytical technique that provides information on the chemical composition of a sample. It involves the generation of X-ray spectrum from the integrated scan area of the SEM, scanning electron microscope, or the micrograph. There are certain energy constraints which are used in this analysis. Now, what is energy constraint? It means something that controls you by keeping within particular limits. This means we will study about the energy limitation that we have to keep in mind while dealing with EDX system. These limits are the energy resolution, that is up to 130 electron volt, which is seen at full width at half maximum. The limit of detection is 1000 to 3000 ppm. The element identified are elements having atomic number greater than 3. The spatial resolution is for low atomic weight. It is 1 to 5 micrometer cube and high is 0.2 to 1 micrometer cube. The precision is approaching plus minus 0.1%. The basic principle of EDX analysis. Energy dispersive X-ray spectroscopy is based on the detection of characteristic X-rays emitted from an element as a result of the de-excitation of core electron holes created by a high energy electron beam. An electron from a higher binding energy electron level falls into the core hole and an X-ray with the energy of the difference of the electron level binding energies is emitted. Due to the quantization of electron energy levels, the emitted characteristic X-ray energies for element will generally be different from the element to element, with only a few spectral peaks overlapping. If the identification of one peak is ambiguous, other peaks or limited knowledge of the sample history will often allow a reasonable element identification of the peak. The normal electron beam of a scanning electron microscope is used as the excitation source. The sample is effectively the anode of an X-ray source. The addition of an energy measuring X-ray detector with a thin, low mass element window on a SEM enables EDS for all elements from carbon on up in atomic masses. A spectrum consists of all the characteristic X-rays emitted by the elements present in the sample on a continuous background due to presimilar X-ray emission caused by the deceleration of the high energy electrons of the electron beam in the sample. The important EDX parameters. The important EDX parameters are: number 1, the count rate for a good quality spectrum, that is good resolution and fewest artifacts. There should be use of 50 to 100 microsecond time constant, that is pulse processing time, with a dead time of 20 to 40% and 500 to 2500 CPS, counts per second. These are good numbers if sample consists largely of high energy peaks, that is greater than 1 kilo electron volt. But if the spectrum is dominated by low energy peaks, that is less than 1 kilo electron volt, then a count rate of 500 to 1000 CPS is better and the 100 microsecond time constant should be used. The other parameter is accelerating voltage. The over voltage is a ratio of accelerating voltage used to the critical excitation energy of a given line of an element. Typically, the overvoltage should be at least 2 for the highest energy line and no more than 10 to 20 times the lowest energy line of interest. The parameter third is takeoff angle. Typically, takeoff angles will range from 25 to 40 degrees. This angle is a combination of the detector angle, its position, sample working distance, and the sample tilt. The sensitivity for low energy, X-ray, and or signals characterized by high absorption can be enhanced by increasing the takeoff angle. Some inclined detectors, for example, a detector angle of approximately 35 degree above the horizontal. Some inclined detectors do not require sample tilt. Horizontal anti-detectors require that the sample be tilted to achieve an optimum takeoff angle. Now we will talk about EDX Spectrum Interpretation. Continuum X-rays as a result of inelastic scattering primary beam electrons in which the electrons are decelerated and lose energy without producing an ionization of the atoms in the sample. Continuum X-rays are formed. The continuum X-rays are the background of our EDX spectrum and are sometimes referred to as the Bresenbler. In theory, the continuum can be expected to extend from the maximum energy of the primary beam electrons and increase exponentially to 0 kilo electron volt energy. In reality, the background goes to 0 at the low end of the energy spectrum due to absorption by the detector window. The detector depth layer and the gold layer, the intensity of the continuum is related to both the atomic number of the sample as well as the beam energy. The continuum intensity also increases with beam current. The characteristic X-rays. Inelastic scattering events between primary beam electrons and inner shell electrons, which result in the ejection of the electron from the atom within the sample and may lead to the formation of a characteristic X-ray. The ejection of the electron leaves the atom in an ionized, excited state and permits an outer shell electron to move to the inner shell. Because the energy levels of the various shells are related to the number of charges in the nucleus, the energy of the emitted X-rays is characteristic of the element. The beam electron must have an energy greater that is just slightly greater than the energy of the shell electron. This is called the critical ionization energy. The depth of excitation. Although electrons may penetrate in specific depths within a sample, which can be illustrated with a variety of equations or with Monte Carlo programs, the electrons actually lose energy in steps as they go to greater depth in the sample. As a result, an electron may soon lose a sufficient amount of its energy such that it can no longer excite characteristic X-rays. Typically, this occurs when its energy drops below the critical ionization energy of the elements in the sample. Each element within the sample will have its own critical ionization energy and its own excitation depth. The ratio of the primary beam energy to the excitation energy of the element is referred to as the depth of excitation. The sample preparation. It is the most important part to prepare a sample for proper analysis using EDX. This means what all parameters one must keep in mind while preparing the samples. So, while preparing the samples, following points must be kept in mind. The sample taken must be stable under vacuum, as the sample chamber is evacuated to prevent the atmosphere from interfering with the electron beam or X-rays. Since EDS is a surface phenomena, so it is advisable to keep the surface as clean as possible, as any impurity on the surface will alter the compositional calculations. Qualitative calculations can be obtained by taking solid of any thickness, but reliable calculations are obtained from qualitative measurement by keeping the sample as thin as possible. So, if the above mentioned points are not kept in mind, there might be chances of contamination and we will not be able to get accurate results. About the data interpretation. Once the sample is prepared, the process begins and there comes the point where we need to interpret the data. The interpretation of data is broadly done by using different techniques like qualitative analysis and quantitative analysis, elemental mapping, and line profile analysis. We will be discussing each one of the above techniques in details in the subsequent slides. The diagram in the slide shows how data is being interpreted. We have samples from which characteristic X-rays are being generated, out of which are further analyzed by a detector, and final data is shown with the help of peaks. To begin with, what we have, the first is qualitative analysis. From the name, we can guess what all we are going to deal with under this analytical part. Qualitative means we are talking about the quality of the material or the sample. In this analysis, the elements that are present in the sample are characterized from their characteristic X-ray peaks. Elements with atomic number from beryllium to uranium can be detected. The variation of detection limit depends on the element and the sample matrix. If there are large number of elements, the peaks that are obtained are overlapping peaks and they are identified depending on their peaks in the different X-ray families. General guidelines for EDS qualitative analysis. Only peaks which are statistically significant should be considered for identification. The minimum size of the peak should be 3 times of the standard deviation of the background at the peak position. P should be greater than 3 b power half. The apparent weights of peak members in a family provide an important source of information in identifying elements. For example, K family, K alpha is 1, K beta 0.1, L family, L alpha 1, L beta 1 is 0.7, L beta 2 0.2, L beta 3 0.008, L beta 4 0.05, L gamma 1 0.08, L gamma 3 0.03, LL 0.04, and M family, M alpha 1, M beta is 0.6, M gamma 0.05, and M zeta 0.06. Begin with the most intense line towards the high energy region of the spectrum, where lines within a family are well separated. If it is above 3.5 kilo electron volt, it must be either a K or L line. Using the KLM markers, compare the location of the peak so that of the marker, check the relative intensities. If you identify a K alpha line, then K beta line should be about 10% and the K alpha intensity. K alpha and K beta lines are typically resolved at sulfur and above. Below the K beta is so small that K alpha and K beta collectively show themselves as one symmetrical peak. If a K line is identified, then look for L lines if they occur for that particular element. If the line chosen does not correspond to a K line, try an L series. If the line is identified as the L alpha, several other lines both above and below the L alpha should be present and correspond to the element. The EDX system should be able to display them at their correct relative intensities. M lines do not exist above 3.2 kilo electron volt, so one would not look for them at this point. Then continue to identify lines while working towards lower energies and consider the possibility of M lines. The lines of an M series are generally not all resolved but contribute to an asymmetrical peak. In fact, M lines can often be recognized by this distortion, but beware of overlap that may look similar. When all high energy lines and low energy L and M lines are identified, light elements can be attempted. These will be K lines and may be overlapped with L lines from heavier elements. If it is not possible to distinguish them, peak deconvolution software or peak stripping may be necessary. Peak stripping is a mathematical process that allows the analyst to remove identified peak structures to reveal possible hidden peaks if they exist. After all major and minor peaks are located, trace elements can be attempted. In this case, problem is to confidently find a peak above background. Only the major peaks of a series will be visible, and that may be lost in a background. In quantitative analysis, from the name, we can guess what all we are going to deal with. Quantitative means we are talking about the quantity of the material or the sample. It can be obtained from X-ray count in different energy levels. On the basis of analysis parameters and the sample composition, semi-qualitative results are easily available without standards with the help of the mathematical corrections. We move on to the elemental mapping. What elemental mapping is? The intensity of X-ray is measured with respect to the lateral position of the sample. The concentration of the element across the surface is indicated by the variation in X-ray intensity. So it is kind of a line profile how the compositional variations are occurring along that line in a given sample. The line profile analysis. X-ray is identified for particular position along the line. By analyzing the X-ray energy spectrum at each position, it gives a plot of relative elemental concentration for each element versus the position. About the window type. In SDD, we have discussed about the window, which is there. So basically, student, these windows are of two types. Number one, the beryllium window type. Number two, polymer thin film window. Let us study about them. Shows a comparison between beryllium window and polymer based window. The beryllium window types, they are highly robust, they strongly absorb low energy X-rays. As a result, element from sodium can be analyzed. The thickness is about 8 to 10 micrometer. The polymer based window, these are less robust, they are transparent to much lower energy X-rays, mainly allowing detection of X-rays down to 100 electron volt. The thickness is less than 8 micrometer. The interpretation of a graph obtained in EDX analysis. A graph of electron diffraction spectrum of Zedite showing the K peaks of sodium, A1 and aluminum and silicon. Let us interpret this graph by taking an example. This graph is an ED spectrum of Zedite showing peaks of potassium, silicon, aluminum. In case of X-ray spectra graph, the energy, which is in kilo electron volt, is plotted on x-axis and the number of counts on y-axis. By examining the pattern of peaks on the spectrum, different elements present in the sample are observed. You can see the relative intensities and the area under the peak are different. The interpretation of the graph. When any sample is being evaluated by EDX, the information is usually represented as histogram or spectrum. Each spectrum is characteristic of a particular element and every element has fixed K alpha, L alpha energy values. For these energy values, the elements present in the sample are identified. In EDS, K alpha emission lines develop when an electron transition to the K shell, that is principal quantum number 1, from a 2p orbital of L shell, that is principal quantum number 2. The line is actually a doublet having slightly different energies depending on the spin orbit energy between the electron spin and the orbital momentum of the 2p orbital. K alpha is typically by far the strongest X-ray spectral line. For an element bombarded with energy sufficient to cause maximally intense X-ray emission. Similarly, L-alpha lines are developed when an electron transition takes place to the L shell from the M shell. In this graph, you can easily see that excitation of electrons takes place, as a result of which the electron moves to a higher excited state, and then after losing energy, it returns back to its original state. The energies are represented by the values of K alpha and L alpha. Every element has fixed values of K alpha and L alpha. Now, next example is of biotile. Biotile is a silicilicate material which is a mica group having formula potassium, magnesium, iron, whole thrice, aluminum, Si3O10 OH whole twice. It is a shell silicate in which iron, magnesium, aluminum, silicon, oxygen and hydrogen form sheets that are weakly bonded together with potassium ions. In biotite, strong peaks of magnesium, aluminum, silicon, potassium and iron are seen. The peaks of hydrogen, oxygen and fluorine are not visible, which shows that the instrument has a beryllium window. The small peak of titanium shows that it may be present in small amount. From the table having values of K alpha 1, K beta 1, L alpha 1, L beta 1, M alpha 1, M beta 1, we can match the energy values and identify the elements present in the sample. The EDX spectrum of biotile is shown above. Students, you can easily see the different peaks of different elements. This is a typical picture of EDX spectra. This is a case study of biotile. Students, in this you can clearly see the different elements are highlighted or they are present. The graph is plotted in counts versus energy in electron volt. The various elements seen are magnesium, aluminum, silicon, potassium, titanium, iron, etc. The periodic table of elements with their corresponding X-ray energies. From this table, you can easily match the different values of K alpha and L alpha and identify the presence of a particular element. Energy dispersive microanalysis. From the value of K alpha and alpha, we can find out the element that are present. As in main EDX technique, it tells us about the various elements that are present, their weight percentage along with the atomic percentage. Now we will talk about the accuracy of EDX. EDS or EDX can be used to determine chemical composition that are present in a sample and also their relative abundance. The accuracy of quantitative analysis of the sample composition is affected by various factors. Many elements will have overlapping X-ray emission peaks. For example, the titanium K beta, vanadium K alpha, manganese K beta and iron K alpha. The accuracy of the measured composition is also affected by the nature of the sample. X-rays are generated by an atom in the sample that is sufficiently excited by the incoming beam. These X-rays are emitted in all directions. This emission is isotropic in nature, and so they may not all escape the sample. The likelihood of an X-ray escaping the specimen and thus being available to detect and measure, it depends on the energy of the X-rays and the composition, amount and the density of the material it has to pass through to reach the detector. Because of the X-ray absorption effect and similar other effects, accurate estimation of the sample composition from the measured X-ray emission spectrum requires the application of quantitative correction procedures, which are sometimes referred to as matrix correction. The strength and limitations. In the strengths we use in spot mode, a user can acquire a full elemental spectrum in only a few seconds. Supporting software makes it possible to readily identify peaks, which makes EDS a great survey tool to quickly identify unknown phases prior to quantitative analysis. EDS can be used in semi-quantitative mode to determine chemical composition by peak height ratio relative to a standard. Now about the limitations. There are energy peak overlaps among different elements, particularly those corresponding to X-rays generated by emission from different energy level shells, that is K, L and M, in different elements. For example, there are close overlaps of manganese K alpha and chromium K beta, or titanium K alpha and various L lines in beryllium. Particularly at high energies, individual peaks may correspond to several different elements. In this case, the user can apply deconvolution methods to try peak separation, or simply consider which element makes most sense given the known context of the sample. The wavelength dispersive method, that is WDS, is more precise and capable of detecting lower elemental abundances. EDS is less commonly used for actual chemical analysis. EDS cannot detect the lightest elements, typically below the atomic number of sodium. For detectors equipped with a beryllium window, polymer-based thin windows allow the detection of light elements, depending on the instrument and operating conditions. So, students, let us summarize what we have studied in this module in analysis of EDX, or X-ray dispersive X-ray analysis data. You know, EDX is an analytical technique that is used for the analysis of an element or chemically characterizing a sample. This is a non-destructive technique and is useful for studying a range of materials. Elements whose atomic number are less than 11 are difficult to be detected by energy dispersive X-ray analysis, as the silicon lithium detector is produced by beryllium window. In order to get the best results, there has to be a balance between the count rate and the resolution limit. By extending the period of measuring each X-ray, spectral resolution can be improved. When two photons arrive at the detector at the same time, then they are not measured as a separate event. Rather, they are added to the energy histogram as one single count, making the resultant peak distorted. Therefore, in order to avoid piling up of the peaks, it is advised to keep the dead time below 30%. EDX has been widely used in material science and nanotechnology and in several biological applications. Although it is not good for low atomic number samples like carbon, hydrogen, nitrogen, but it is still widely used for biological applications as well. Although new analytical techniques have come into existence, EDX is firmly established as a standard microanalytical technique and will remain at the forefront of the microanalysis. Thank you so very much.